Thanks for coming back, and I hope you all enjoyed a break from the sessions and were able to check out the exhibition hall and Spud Shed. A reminder that by visiting those, you will gain points which will give you the opportunity to win prizes. For those who are just tuning in, my name is Ashley Robinson, and I am the editor of Spud Smart. Also, a reminder, if you have any questions for our presenters, please make sure to type them into the chat box and our presenters will reply to you. Our next presenter is Mark Stahlhelm, who will be speaking on the use of cover crops in potato rotations in the United Kingdom. Mark has his own consulting company, Mark Stahlhelm Potato Consultancy Limited, which he just started last year. Previous to that, he was head of the NIAB CUF Potato Research Group, where he guided the overall scientific direction, development, and management of the group's activities. Thanks, Mark, for taking the time to be with us today. Please take it away.
Good afternoon. My name is Mark Stahlhelm. I thank you for the invite to come and talk at the Canadian Spud Congress this year. Very quickly, I'm going to give you a background of what my role is. Until November last year, I was head of NIAB CUF, a potato research group based in Cambridge in the UK. I've now sort of moved over to the dark side of being a private consultant after 34 years of work in the research industry. I did my PhD on crisping quality and the effects of site and season on that, and really my subsequent research has focused primarily on soils and water. So today, in response to the organisers, I'm presenting the findings of the AHDB Potatoes Rotations Project, and the AHDB is our national levy board, and work on the effects of cover crops and soil amendments with my colleague Mark Allison. That's a 5-year project, and I'll report very much on year 4.5 to 5 as where we are now. Also, what I'd like to touch on is the work of biofumigation by Bill Watts, who submitted his PhD 2 years ago. So, benefits of cover crops. Very basically, we have soil stability and nutrient capture. We have basically the building of biomass and incorporating it into the soil so that we get an improvement in organic matter. The other one is biofumigation, as I touched on, and we'll go that immediately after this slide. The other one is the effect of using tap roots or more vigorous rooting varieties to overcome compaction in the soil. So rooting, natural processes, rather than actually using steel to overcome compaction. And the other one is where I'm focusing on is soil drying and the aid to spring cultivation, because potatoes are planted in the UK in March, April, May typically, and therefore we've got to deal with soil that's come through winter. So let's start with biofumigation. Very simply, biofumigation is a technique which uses volatile organic carbons, so VOCs effectively, which are released from glucosinolates, which are contained in brassica tissues. So the little diagram you've got there shows a glucosinolate molecule basically combining with water and catalyzed by the enzyme myrosinase to produce these isothiocyanates, ITCs, which we'll talk about later on. So these are within plant cells. So within plant cells means we have to get them out. So we'll talk about the way we get them out and into the soil to control the soil pests. So I mentioned Bill Watts. He submitted his PhD thesis. He worked for the levy body for a number of years and he's now working for another independent agronomy company. But I'm very thankful for him to be able to present these few slides on his work. So very quickly, it's the glucosinolate profile contained in the cover crop that we're growing, and we'll come over that in the next slide. The quantity of biomass. So very simply, we have a concentration in the cell, and then we have biomass. So you'll see there that the concentration times the biomass is crucial to get the kill that we need. Plant physiology at maceration. Have we got lignified stems? Have we got fresh green material? What's the state of flowering? Have we got the right concentration? And the other one is it's fitted in a rotation graph, and you'll see where we need the soil water and the temperature right. Then we need to be able to plant it in late summer to get our fumigation in early autumn and therefore get the control that way. So the speed of growth is very important, and the maceration and incorporation of that ideal biomass, so concentration and biomass there, depends on the implement we use. So going through there, you can see there the three that I've highlighted in terms of ITCs are propanol, benzyl, and phenyl ethyl. And those are the three common ones that we get the most efficient kill of globular rostockiensis. So PCN in our case, not root knot nematode, this is what we're dealing with as this particular pest and targeting. And that's Bill's work's focus. So if we're asking chemical companies or seed companies, we're looking for basically more than 100 micromoles of ITCs in the tissue of the plants that we're growing in terms of, glucosinolates. So very simply, as I mentioned, it's mass times concentration, low mass, high concentration, high mass, low concentration, but ideally high mass and high concentration. So we need to reach that flowering just beyond to get that maximum stage. And you can see there the three different species that we've got there, the biomass in fresh weight, 25 to 80 tons for some of the crops, and the particular ITCs that are controlling it. But we've got to grow it as a crop. We need nitrogen and sulfur to give us those high glucosinolate levels. So we have to fertilize the crop, and we may need to establish it with irrigation if the soil is dry. So very simply, expediency. The ITCs, some of them persist for as little as 30 minutes. So we've got to basically get them in the soil out of the plant material, seal the soil so that we can prevent them going, and we need to get them volatile. So ideally, we do that in a very quick one-pass operation. Occasionally, we might need to do it as two passes, but time is of the essence here. So simple pass system there. We've got a mower and a macerator on the front combined with a rotavator on the back to give us that incorporation. Here, we've basically got the roller techniques. You'll see some of the data. So the picture on the left is a crimping roller. It's squashing the stems, whereas the picture on the right is clearly there capable of actually cutting the stems. And we really need to do both chop into small chunks and crush to liberate the ITCs as effectively as possible. So here we're going into some results from Bill, and you can see there basically if we concentrate on the lowest bars here, which is the one labeled 53 in terms of the viability. So the lower the number, the greater the kill. So 53, 56, 57 here for flail treatments compared to the roller. The roller, not always significantly better, significantly worse, but numerically over the treatments in terms of whether we spade, whether we plow as a subsequent operation, or whether we rotivate, the flail is giving us better control. If we move on now and just average those data, you can see there, that's the average of all that previous slide, that the flail is significantly better in terms of controlling the PCN that we've got in the soil. And what we're aiming for is basically volatility. So we need 10 degrees minimum temperature to get that volatility of the gas to spread throughout the top 30 centimeters of soil where we're incorporating the organic matter, and aim for somewhere in the region of 25 to 75% field capacity. Quite a big range, but the bigger the crop we have, the more wet the soil needs to be to actually give us the control there. And you can see the results there in terms of the optimum. So that's biofumigation. Very simply, it's work presented that Bill did. It's about getting everything right. It needs to be almost the perfect storm to get everything right. Well, does the same apply in terms of when we go other cover crops? Do we need to get more things aligned than just simply growing it and hoping that we get the consequences of the cover crop? So a series of trials here as part of the rotation program, I'm going to go into in detail, in sort of a deep dive, where we've grown oat cover crops at our base at NIAB over the period 2018, 19 and 20. These are commercial oats. They're not black oats in terms of biofumigation effect. And they contain no admixture, so no legumes for basically our regulatory payments that we receive from Europe, no longer. But basically, if there was any weeds, we did get some weed level coming through into the oats. Moderate to low seed rate, 70 kilos per hectare, not as high as a commercial rate, but enough to give us the ground cover. Drilled rather than broadcast, and then no fertilizer until we actually applied fertilizer for the potatoes in the spring. So you can see here, I'm going to show you some pictures over the 3 years of the project. In 2018, we got some very sparse crops, but I'll show you a slide in terms of soil conditions a bit later, where we got some real benefit in terms of this year. 2019, a much bigger crop. You can see there where the tractor's mowing it. That's our surrounding field where we do our experiments. And you can see in the foreground, the cover crop experiment. So we're actually having to mow it and then plow it and incorporate it in 2019. In 2020, you can see a little bit earlier there, but you can see the lush green cover of the crop. It then turned quite cold and the crop didn't grow much more. So what does that mean in terms of data? Well, we've got our 3 years here. The fields are separated no more than a few hundred meters apart in terms of distance. But you can see there that the dry matter yield and the nitrogen content and the carbon to nitrogen ratio vary considerably across those 3 years. So 2018, 2020, we got similar levels of dry matter yield, sort of 2 to 3 tons of dry matter. The nitrogen content in kilograms per hectare in the 50s, whereas in 2019, we had a crop that was almost 4 times as big in terms of dry matter, and its nitrogen content was about 2.5 times as great. So very variable results in terms of that. And obviously the carbon nitrogen ratio, the one in 2019, despite its high nitrogen, was more lignified. Does that make a difference in terms of its benefit to being incorporated? So you see the problem we've got here is we've got 2 superimposed pictures here. The bottom is a cover crop that was burnt off very early on in the season, and you can see there it's dead, and we've got down there about probably in the region of 11 inches or 27 centimeters in terms of depth of soil to ploughing. And you can see on the top there where we've got a growing cover crop, whereby it looks and appears a little bit dry at about 6 or 7 inches, 15, 20 centimeters, but actually deeper down it's still wet. And so what we're trying to do is gain the benefit of that evaporative surface you can see in that top picture, drying the soil in our traditional cultivation period, which would be late March, early April. So if we look at this, there's quite a lot of data on here, but I'd like you to focus, like on the other slides I'm going to show, on the red numbers to highlight some differences here. So we've got here some dates, basically 11 days apart, late March in 2019 to early April. And we can see there that the surface layers on the surface actually were drier where we had no cover crop, because we've got wind effects. The effect of having a cover crop is to provide a mulch and prevent soil evaporation. So we've actually got drier soil in both instances in March and April. But if we look at depth at 30 centimeters, we can see there the soil is above field capacity. It's about 27, 28% in terms of field capacity at Cambridge in this field. And we can see there all of those growing treatments were actually at field capacity and above. But if we move through to the 1st of April, we can see the 2 treatments that basically had no cover crop over the winter, or one that was defoliated much earlier, actually having a soil which was still on the boundaries of field capacity, about 27%. But the growing crop actually had now dried the soil at 30 centimeters depth. Therefore, we're able to cultivate our sandy loam, sandy clay loam soil without fear of a plastic limit being exceeded and getting shearing compaction at depth. Ideally, we would have delayed another couple of days, but my colleagues were pushing me to go on, and so we decided to plough on the 1st of April. Ideally, we would have allowed 2 more days. If we look at the bulk densities, the first set of data that we can see over here is basically there's a trend. It's not significant, but there's a trend for the bulk density to be lower under the crops that had cover crops over winter. But if we look particularly in 2018, the previous year, where soil conditions were much poorer, as I will show you, we actually did get quite significant effects of the cover crop in terms of reducing bulk density, improving porosity, improving drainage, and improving rootability. So if we move now forward to last year, 2020, you can see here we're a little bit later. We allowed the cover crop to go on much longer in the experiment to see how much soil water we could actually drive off. And you can see here, we're in a lighter area of the field because the soil water contents are lower. But actually we're seeing there that at 30 centimeters, you mentioned the previous slide where we saw 23 or 24% by early April, we're now down at the level of about 16%. So we've got significant drying at the depth we want to do our primary cultivation. I'll show you the tool that we used in a moment. But the 2 pictures you can see on the right where the soil is on a spade are effectively dug out at the depth we want to do our primary cultivation. So we've got a string there. It's about 6 to 7 millimeters in diameter. So it's on that bordering plastic limit where we want to cultivate. So we would get compaction if we cultivated that soil at that particular depth. The soil on the bottom is the one where we've had a crop that's been grown right to the point when we're going to do our primary cultivation. So you can see how friable it is there at that particular stage. So I said I'd return to this soil. This is 2018, the first year of the experiment. The soil that was dug out of the crop on the 9th of March 2018 came out of the pit exactly as you see here. And so effectively the crop that was grown into soil, the potato crop that was grown into the soil on the left, had a cover crop growing the entire winter from September right through until this point in early March. The bulk massive soil structure that you see on the right came out of an area that actually been burnt off, desiccated with Roundup, as soon as it emerged in early October 2018. So effectively, they have no cover crop growing for the whole season. That's a massive difference in soil structure. We got very excited by seeing this. So that year we didn't do it, but we basically followed on for the previous year, the succeeding years of the project to look at VES, which is a visual evaluation of soil structure. And you can see the categories here. 1 is friable and crumbly. 5 is massive and bulk structure. What we're looking for is on our cultivated soils somewhere in the region of 2s and 3s, and you'll see that in a moment. So if we look at our VES scores in 2019, the middle year of the project, actually where we dug a soil out from the surface down to around about 20 centimeters, our cover crop had a slightly better VES score. So they weren't great. I said we're looking for sort of 2s, ideally 1, but we've got numbers there that are right in the middle. But you can see there's very little change over the course of the season until we get close to March, when we see a small drop in here. But soil conditions were not great in 2019, but we'll move on to 2020. 2020, what we find now is actually by the time that we move right through to the time when we're doing primary cultivations at the end of April this year, as opposed to the beginning of April the previous year, we find that we got a significant improvement over time. So moving from really quite poor soil conditions, region of 3s to 4s, moving right down to in this category of about 1.5, which is very good soil structure. And I showed you in a slide what this actually looks like. But if we defoliated early and basically allowed the crop to die as a course of the desiccant, what we found was that the soil structure over that top 20 centimeters of soil actually didn't improve as much as where we had a cover crop still growing. Interestingly, the undefoliated cover crop dried from the soil surface downwards, and did actually give us an improvement there. So what we've got there is some contrast. They seem to be a bit contradictory over years, but that's generally what we're finding is soil structure does depend very dramatically on the season in which they're growing. So if we look at one of the more consistent results is that we've done 24 different trials, split field trials, as well as detailed experiments like I've just shown you. But we actually found that on these trials that there's a significant improvement in bulk density, i.e. a lowering of bulk density in the upper 10 and 20 centimeters of soil, and slightly down here in terms of the 30 perhaps, but certainly the top 2 layers of soil in the cover crop reducing bulk density, so improving porosity, improving drainage. If we move forward now in terms of the ability of the soil to sustain damage from rainfall, so effectively aggregate stability, dunking soil into water repeatedly mimics rainfall or irrigation. And what we find is that if we look at cover crops, there's no overall effect. And we found equal numbers of positive, i.e. there was an improvement in aggregate stability, or an equal number of basically poorer results where cover crops actually reduced the level of aggregate stability. Contrast that with amendments, so compost or farmyard manure that we put on, there was a positive effect there over most of the trials. It was small, but it certainly improved it. So cover crops aren't giving us the aggregate stability we thought might have happened. So how do the crops perform growing into those cover crops? So potato crops planted, and we're going to use 3 pictures of ground cover here in the next 4 slides. 2018, very, very simply there, all 3 treatments there, no cover crop, sprayed off early and then left undefoliated until our primary cultivation, all showed very similar ground covers. And whilst the no cover crop did senesce slightly earlier, it's not great in terms of the overall area under that ground cover curve. Look at 2019, there certainly is no difference there in terms of overall ground cover, but we'll look at the yields in a moment. If we look at 2020, there's some very contrasting effects here. The green line you can see there was where we defoliated the cover crop 6 weeks ahead of any primary cultivation. So effectively, you'll see a picture in a moment, the cover crop was just a mat of dead oat tissue, and it had certainly done something in terms of advancing emergence. Question, was it depth of planting that we differed? Everything is hand planted in our trials. The planting depths were identical across all those 3 lines there. So it wasn't the fact that the crop was planted slightly deeper while we got that early emergence. Something was going on in terms of the soil structural conditions or the biomass was incorporated, or the dryness of the soil that advanced that emergence. And so, not surprising, advancing emergence, the red line in this case is the early defoliation. So we get earlier ground cover, and the crop actually doesn't senesce any later or earlier than the other treatments there. So there is a movement there to earlier ground cover. 2020 was a very bright early season. May was the brightest May we've ever recorded at Cambridge. So early ground cover would have been beneficial in terms of improving yield compared to slightly delayed. So if we move on to the yields, there's 2018, 19 and 20 there, and I've highlighted the red areas where there are significant differences either in total yield or marketable yield there, and that's greater than 40 millimeter size. In 2019, there were no overall effects. Numerically, the undefoliated had the highest yield here, but there were significant effects in both 2018 and 2020. So in 2018, we found that the defoliated crop, so that was one that basically was defoliated about 6 weeks before we did our primary cultivation, had the lowest yield. But there was no difference between having no cover crop and having undefoliated, despite those differences in soil conditions that we saw between the cover crop here, which was poor, and this was good. We still didn't manifest that in terms of yield. But defoliating late, where we actually get a mass of green material that dies, actually prevented that soil drying out, and we did end up with a lower yield as a consequence. So moving forward to 2020, where there are again significant differences, the defoliated early, which is the one that emerged earlier, as I showed you, actually did have a very significant increase in yield. So at the order of 6 or 7 tons per hectare increase in yield. And certainly that's a major advantage in terms of the cover crop. So we see effectively no effect, an effect of a cover crop being neutral, an effect of cover crop being positive. But when we burn it off, does make a difference. So the management of getting this right to optimize yield seems to be pretty tricky. So if we move on, effectively what we've got here is 2020 crop, and you can see I'm going to talk about the cover crop in its incorporation now. This should be a little video that comes up which will show you. If we move on and look, effectively the picture on the bottom is where we defoliated the cover crop 6 weeks earlier with Roundup, and on the top we've got that fresh green crop that has actually been not incorporated. But one of the techniques we use in the UK is to destone soils in potatoes. We have stony soils or even declod. And I'm going to show you a picture of where these two particular examples of cover crop residue might cause a problem. So if we move on here, we can see the destoner. What it's doing is effectively sieving the soil from a bed of potatoes and dropping out the clods or stones into an alleyway here. You can see these little pieces of straw. So this was a stubble, not a cover crop prior to planting. And you can see if I move on here, what sort of level of debris we're getting out. So if you can imagine that sward of oats coming out there, it's clearly an issue there that we might either take it out to the side into this area of furrow. We actually might get blockages, and many growers talk about blockages in terms of. So what we'd be looking for is managing that. So if we move on now, we're going to look at some commercial trials where the results of some of these findings have been put together. Well, you can clearly see this is pre-2018 when we did the work at Cambridge. So all along our route, we've been looking at how we can improve it. And cover crops, some growers have taken up and adopted quite rapidly during the phase of the early part of the project. So you can see here that an agronomist called me out having taken some samples for looking at nutritional analysis. So you can see where he's mown the crop there to take out the nitrogen analysis of the crop. But he did call me and say that soil conditions were extremely good on the area where cover crops were grown, whereas where they'd been defoliated effectively on the 8th of March, they were very, very much different in terms of structural stability. Now, what I got him to do was to take some soil samples, bag them up, seal them, and we measured their water content. And overall, from a depth of the surface down to 30 centimeters, the soils that have been growing the cover crop actually had about 1% lower water content than where we desiccated on the 8th of March. So what we've got there is quite a significant effect, but is 1% really that important in terms of overall? Well, I'll show you the results that we got as a consequence. So you can see the picture here where we've got the variable soils texture up here, or in this area where the red square is, which is basically a sandy clay loam, but variable, as you can see. The trial you can see here is situated in the sort of yellow area that we can see in the bottom here. And the line you can see almost down a diagonal line across here is from the area where the cover crop was desiccated with Roundup on the 8th of March versus where it was actually not desiccated at all, so undefoliated. You can see there's a clear difference in soil texture across here. Now, you might think it's variable. Actually, these two textures in the area of our experiment were identical. So we were in a reasonable area of uniformity in terms of soil texture. But what we followed was the tractor drivers all measured their works of rate. So the ploughing, which was across the field, measured his work of rate and was about 22% faster where we hadn't defoliated the crop. The next operation to bed form, to put those beds up before we destone, actually was about 12% faster. The bed tilling operation, which is a way of getting rid of these clods prior to doing the destoning operation, which is the sieving of clods and stones, was 19% faster. And the destoning operation, the last in the line before we plant, was actually about 24% faster. See, overall, a significant improvement in work rates with a crop left to grow right until the point when we ploughed it in late April. So we're seeing these results. Well, yeah, that's savings, improvements in soil structure. In this case, because they were strict trials, we can't compare between those two lines of data, but we can look at inferences. Would they be different or not? So we can look at yield and the total percentage of yield above 90 millimeters. This was a processing russet Burbank crop. You can see improvements there in terms of overall yield. We can't use statistics to analyze them, but if we do a lot of this work, we generally find that cover crops can improve the overall yield, and I'll go through that a little bit later. So more work rates, more data. Here you can see a fodder radish crop grazed in the southwest of England, basically received a dose of Roundup in May 2018. It was topped, and then the cultivations were done about 4 days later. So the primary cultivation here, the red numbers again, pay attention to those. With the cover crop, almost a halving of the fuel consumption to produce the initial seed bed prior to bed tilling and destoning. The work rate slightly faster. The bed tilling, it's a very intensive rotary operation, consumes a lot of fuel, as you can see here in terms of litres per hectare. With a cover crop, significant reduction in fuel. Work rate's not changed, but a reduction in fuel. The destoning, basically no real change in terms of fuel consumption or work rate. So we move forward now. We've talked about soil structural conditions. This is leaving cover crops to grow over winter. So drilling in September and then going through right until April when we would do our primary cultivations. Well, what about the benefits of growing a cover crop perhaps a little bit earlier, but taking them out significantly earlier, i.e. pre-Christmas? Can we get that benefit in terms of overall and performance? So we've got here basically mustard or radish crops growing. You can see the strip that we've basically not drilled in the middle. So that's our comparison, cover crop versus no cover crop here. We've done a number of these, as you'll see in a moment when we talk about the number of trials we've done, simple strip trials, comparison of with and without cover crops. So we move forward here very simply. Here is again the nutrient uptake in terms of nitrogen, the cover crop dry matter yield. And again, pretty much like we had at Cambridge, 2 years with moderately low, sort of 4 to 5 tons of dry matter per hectare, grown and then cultivated in either in January or late October. And then if we look at the N uptake, somewhere in the region of 111 to 130 kilos of nitrogen. But the year in the middle, 2019, we saw a very big increase in the cover crop yield, despite a moderate incorporation date here in early November. And then we look at the cover crop N uptake, it's about 230 kilos. So effectively a very big difference. So very big differences in nutrient uptake and biomass produced from 3 very similar drilling dates and very different growing conditions during that autumn or fall period. So we look at the yields there. Again, concentrate on the red number because it's the only significant difference we've got there. I'll come back to that in a moment, why we think it's significant. But you can see there the overall of all those is if we take all of those numbers and divide by 3, there's a benefit in this particular grower of growing this mustard crop in the period of fall of about 2.2 tonnes per hectare. So remember that number, about 2.2. Now, we've done this now as a result of the project, basically looking at 27 cover crop trials with and without. And this graph shows the little blue bars on top of the white bars are actually the effect of the cover crop. So down here, we've got 1, 2, 3, 4, 5, 6, maybe 7 crops where there is a yield disadvantage actually compared to not growing a cover crop. But if you look at the number of crops where the blue bar is above the white bar, these are all improvements in yield. They might not all be significant, but they're improvements. So if we look at here, the number we've got, when we look at the final bar here, it's the average of all of those 27 crops. And if we look at that number, we'll come to it, what it means in terms of numerical. So the height of that blue bar in a moment. Now, those are experiments or trials. What we've also been doing is collecting survey data from the same growers in the rotation project. And basically what we've been finding is that if we grow a cover crop, there's significantly more there. We've got 30 grower instances of no cover crop in our survey. We've only got 14 with cover crops. But very simply, we can find that the mean benefit of potato yield is higher where we're growing cover crops. So we can look at that difference there. It's in the order of 54 tonnes here as an average and about 44 tonnes where we don't. So there's a lot of factors influencing here. Do people that go cover crops, are they generally on better soils or are they in different soils? Do they have livestock? Are they better growers? Do they have shorter or longer rotations? A lot of error in here. But as we get more data, we'll be able to reduce the confidence level of here downwards. So these are the extremes of the range. But nevertheless, that's the benefit. But if we look at organic amendments, compost, cover, farmyard manure, and other bio amendments, what we can see there that there's very little effect on mean effect in terms of overall improvement. So if we combine all of our organic matter experiments and trials, so not the survey work, we find there's a bit more of a mixed picture in terms of there are more crops where we see basically a disadvantage in having organic amendment and some where we see the benefit. And overall, you can see the little height of this bar here is an average of all of those 28 trials. So if we summarize that, for cover crops, basically the use of a cover crop increased potato yield in our experiments and trials by about 2.8 tonnes per hectare. So I told you to remember the one where we looked at the one grower, his was about 2.2 tonnes. And a paired t-test over all of those trials of cover crop versus no cover crop showed that this increase was significantly different, so around about the 1% significance level. In contrast, adding an organic amendment to the soil, so farmyard manures and compost, improved yield by slightly less, but overall those 28 trials, we haven't seen a significant increase yet. So there's too much noise around those data. We look at here, obviously it costs money to grow a cover crop. I put them in Canadian dollars for you. So you can see an average there is about 370 Canadian dollars to establish the crop. So that's basically establish it and kill it and incorporate it. So you can see there's quite a big cost saving there. If we summarize then, so if I summarize the last bit we've been talking about first, which is the average yield benefit from soil conditioning cover crops is about 2.8 tonnes per hectare. That equates to about 875 Canadian dollars, and the cover crop cost variable, but they're averaging about $370 per hectare. Big range in growing the cover crop, obviously. We have improved cultivation speed despite those extra crop residues, and it's trying to convince growers about the management at the end is we need to chop and incorporate that material, and then we can handle it on the destoner. We don't need to have big swaths of unchopped material. Don't overwork the soil. If it's in better conditions, we should move faster, not create finer seed beds by going at the same rate. With soil structure, we've seen reductions in bulk density consistently with growing cover crops, and the VEST scores generally are better, but there is quite a lot of noise in the data. However, there's little effect on water-stable aggregates, so organic amendments do appear to be better in improving that system. So we'll come back to management right at the end, but biofumigation, we need the perfect storm. We need everything aligned. We need to select varieties of cover crops which are rich in glucosinolate, particularly of those particular glucosinolates there. They've got to fit in the growing window, so late summer through to late fall, and produce biomass rapidly. So it's concentration times mass that we need. We need to macerate, we need to chop, crimp, and roll rather than actually just, chopping in big long lengths or ploughing under. We need to have the soils warm and we need it wet. And so that's the key, is getting those optimal soil conditions. And costs, well, they're pretty high, but what price do we have when we're losing nematicides and nematostats day by day almost? So $300 to $600 per hectare. So as a summary, managing cover crops is the real key. So there's a lot of time and effort involved, but done right, we can actually see benefits in terms of yield. The question then comes in about what other pests are we perhaps harboring in our cover crop, where we're growing a crop where we might not be growing anything. So thank you for the invitation to speak. I would welcome any time we've got for questions, and I know there's a sort of breakout session at the end where we're all invited back, and I will be there at that time. So thank you very much.
Thanks so much, Mark, for that informative presentation. I know how interested growers here in Canada are about cover crops and how they can help their crop production.